First of all, I would like to thank the organizers of this conference for the invitation. It was with great pleasure that I accepted to be here and to talk a little bit about what I'm working at. And I will be talking about the introduction of crops in the Iron Age in the Roman period in Iberia, exploring the differences between the two major regions, and how these relate with historical and environmental specificities of these regions. This presentation will follow a rather simple structure. I will start by explaining some contingencies of the data set I will explore, mostly regarding preservation issues, which can somehow promote a bias with interpretative implications. And then I will talk a bit about the introduction of crops in Iberia in the long term, considering the fact that the Iron Age and the Roman changes in agriculture can only be understood if we take into consideration the background in which these changes operated. Is that long term that we were talking about. And afterwards, I will address the Iron Age and Roman crops by identifying the main changes and addressing some relevant sites, and then by comparing the Mediterranean and the Atlantic. And by the Atlantic here, I will assume to be the Northwest Iberian areas. First, we start by referring to the nature of the data set that I will focus on. Ancient communities use very different and abundant plant resources on their day-to-day activities, but only a few will end up integrating the archaeobotanical record, and this has severe interpretive limitations. Although plant remains can be preserved in several ways, carbonization is by far the most common way of preservation in Iberia, and all interpretation must take into consideration the possible biases created by this way of preservation. So, although some tragic events may lead to the destruction of massive quantities of remains, for instance, when a storage area gets burned, these events are rare, and most shared plant remains derive from small accidents in day-to-day activities, since people would not burn food on purpose, most of the times at least. And so the most common way for cereals, for instance, to get carbonized is for the post-harvesting byproducts like straw and so on to be used as fuel in domestic fires. So you can imagine, for instance, a batch of straw coming from the threshing floor and used as kindle in some fireplace. So the hearth or the oven or the fireplace would be cleaned every day or every 2 days, and the remaining ashes and charcoal and so on would be tossed into a dump. And after several weeks or months, or that dump would include several grains that were transported by accident in the middle of the straw and got carbonized in the hearths and in the ovens. So we would get some grains, but probably we would not get any fragment of straw, because straw is much more fragile than the grains are. So this is how preservation bias occur. On the other hand, since people would heat, for instance, they would heat grapes with seeds, a lot of these fruits could have been consumed in that particular site, but not a single seed got burned, not a single seed would be preserved until today. So, so first of all, only the material that gets in contact with fire can get the chance of being preserved. But fire is also a destructive element, so only the most resistant elements survive combustion, which excludes a lot of the most fragile plant parts these will simply get destroyed and turn into ashes. And so all that never gets in contact with fire simply rots away and disappears. So, and that, as I say, the fire usually occurs on domestic areas. So we more easily get them associated with dumps or with fire or ovens, arts, some kind of structures where fire is somehow managed. If you're lucky enough, you will have a waterlogged site in the area. So waterlogged sites usually provide a much higher diversity of plant remains that includes much more species, but also plant parts like pods and so on that usually you don't get in the carbonized assemblages. So usually waterlogged plant assemblages include species that are uncommon or even absent in the carbonized assemblages. So if you have both of them, both types of preservation in your study area, that would be great. But usually that's not what occurs in Iberia. We have very, very few waterlogged sites. And you can see this. For instance, this is the data set, the sites, the Roman sites with carbonized fruits and seeds. Sorry, with fruits and seeds, not all carbonized. The red dots are the waterlogged sites. So very few sites in Iberia. And they usually are associated with, they are coastal sites, some of them, some are not coastal sites, but associated with wells or other structures that contain water. So, but most of the remains are sharp. What we get also from this map is that there are high discrepancies in the concentration of the sites. You see, there are a lot of sites in, for instance, in northeast Spain, and you have a lot of sites in northwest Iberia, northern Portugal and Galicia. And this is basically related with research history. You have strong groups in eastern Spain connected with Girona lab and Barcelona, Valencia, and so on in eastern Spain. And you have our lab of CIBIO here in Porto and the group of my colleague Andrés Teixeira that works in Santiago de Compostela. So this is basically that we have this problem. You know, research history. It's an issue that we must take into consideration. There are huge gaps in this map. These are the Roman sites. I don't have a detailed map on the Iron Age sites. I'm sorry, but the same happens. You have a huge concentration of sites in northern Portugal and Galicia, as we will see, and you have a lot of sites, again, in Catalonia and Valencia region, and a minor concentration of sites in southern Spain. So this geography of investigation in some way is a limitation to our studies, but that's what we have, that's what we can manage. Still, there are some syntheses that you can find in the web. There are some studies focusing on Roman and Iron Age topological assemblages that you can read. You can see that and characterize in more detail the assemblages that I'm talking about. Okay, I had the pleasure to author or co-author some of these papers. Some are quite interesting. I hope you manage the time to read some of them. So the synthesis I'm presenting will focus on these two periods, on these two major assemblages of sites with crops. But I will go back a little bit and starting from the start. I will start from the beginning of agriculture in the Iberian Peninsula. And this is important because most of the species that we will find in the Iron Age or in the Roman period reached the Iberian Peninsula already in the Neolithic. So we need to have this long-term scenario in order to understand it. So the beginning of agriculture in Iberia occurred slightly before the middle of the 6th millennium BC, and it included all the package of domestic cereals and pulses that are spread out on the Mediterranean, as well as flax, and it would later include the poppy. So the cereals are mostly good and free threshing varieties of wheat, and also wheat and naked forms of barley. So this marks somehow a difference between Iberia and the Eastern Mediterranean. In fact, a recent analysis suggests that during the neolithization process, there was an increasing presence in free threshing cereals from east to west. So that is a preference. There is a preference in the older and eastern sites. There is a preference for hammer and hincorn, that is Triticum dicoccum and Triticum monococcum, and whole barley, Hordeum vulgare, while in the west, in the most western sites, there is a preference for free threshing wheat, mostly Triticum durum, but also Triticum aestivum, and the naked forms of barley. So this would continue in most of the Iberia for several centuries after the Neolithic. And only later on in the Bronze Age, there is a new relevant crop being introduced in Iberia. So these are, in fact, there are two crops, two millets. One is broomcorn millet, that is Panicum liliaceum, and the other is foxtail millet, that is Setaria italica, you see here in the picture. They were domesticated in China 8,000 years ago. They only reached Europe more or less 3,600 years ago. This is a new chronology, considering that there were previous studies that suggested that millets had arrived Europe already in the Neolithic, but in the last 10 years or so, new radiocarbon dates has pointed out that it only reached Europe probably in the Bronze Age, and that's what all chronologies are pointing out now. And even even here in Western Iberia, chronologies suggest that we only have one oldest site, probably, but without radiocarbon dates, so we would need more radiocarbon dates to attest the chronology of these sites. So most of the sites are already from the late Bronze Age, which means that they fit in the general chronology of introduction of millets in Europe. So during the Bronze Age, there is as I mentioned, we have wooled the naked forms of barley, but in the Bronze Age, it seems that the naked forms of barley that were predominant in the Neolithic and in the Chalcolithic become less relevant, while wooled barley is increasingly present throughout the whole Iberia. So there is this kind of replacement that we still don't understand why it occurred, but it occurs. So these are the main changes before the Iron Age. So when we reach the Iron Age, we have all these crops, but the fact is that the naked wheat and barley that are here since the Bronze Age, since the Neolithic, sorry, they continue to be probably the most relevant crops in the Iberian Peninsula, well, at least until the Roman period. So with minor inclusions or with some specificities in some particular areas that we see soon. So going into the Iron Age crops, the major shift that occurs in the Iron Age can easily be understood by looking at this particular site in the city of Huelva. Well, you have, again, the major cereals that we have in Iberia since the Neolithic, and they are predominant in, well, at least in what cereals is concerned. But what changes in this 9th century BC site is that we have a lot of different new crops that are fruits and fruit trees. And this is the major change that occurs in the Iron Age in southern Iberia. So we find the core cereal species, but plus a good diversity of fruits. These are relevant. They are present in a particular area of the site. This is in waterlogged conditions. So only in waterlogged conditions we find this diversity of remains. These include some species that are originally from the Near East, such as almonds, pomegranates, and melon, but also indigenous species, fruits, whose cultivation seems to have started by this time, like grapes and olives and figs. So this introduction of new crops and the expansion of these ones it's a very particular and very relevant thing that is occurring by this time in southern Iberia. And you can see it also here. This is also from Huelva. These are vineyard trenches, that are really incredible, and they document the cultivation of grapevine in the 9th century BC. So, comparing this site with others from southern Iberia, it seems there was an expansion of fruit trees from what is current-day southern Spain into the east in the following 3 or 4 centuries. So it happens with figs and olive oil and almonds and pomegranate, and mostly with grapes. So it's something that is occurring from west to east, which is interesting. And but we don't know what is happening more to the west. In Western Iberia, western from Huelva. We don't know quite well what's happening. We have this very interesting site in Portugal, Castro Marim, that is very close to Huelva. This is a 5th century. There is a 5th century level with a lot of amphora that are broken in situ, and these vessels are originated in the Guadalquivir region. They are filled with barley grains and some grape pips, and they demonstrate these regions are in close contact. In fact, the contacts are clearly testified by lots of ceramic assemblages as well as other materials that are found along the Atlantic coast. So this is a completely new world. And of course, this happens as a consequence of the colonial contacts with Phoenicians. So it is secure to say for now that fruit cultivation expanded in the southern areas of Iberia under the Phoenician influence. So, and Huelva and the presence of this particularly relevant site in Huelva is something that you can easily understand due to the fact that this was a very relevant area in terms of Phoenician colonization. So this Western-Eastern movement is probably a result of the relevance of this bay during the Phoenician colonization of the Phoenician contacts in Iberia. What happens afterwards, after the Roman conquest, is a change in the production strategy and scale of the production, of course. There was probably some 50 years ago, there was this idea that there was some agricultural colonization of Iberia in Phoenician times. This was dismissed afterwards through several investigations by many Iberian archaeologists. What we find is that there's the major change in scale of production occurs at Roman times. So production becomes massive, and in some sites, it's almost based on extended monocultures, particularly in southern Spain, in the province of Adelaide. And the most common cultivated fruit trees that we have in the Mediterranean sites in Roman times are figs, grapes, and olives. And only in this period, these three species become particularly dominant. What we see is that, for instance, that olive oil in Andalusia, from the Roman period onwards, is spread from the Thermo to the Meso-Mediterranean zone, and this is clearly evidenced by charcoal data also. But what archaeological information testifies is for major changes in landscape and site distribution. We have an abundance of villas and farms directed to the production of olive oil and its transportation to the whole empire. And it is absolutely impressive. It's all over the place. So this is something that we don't have. We don't have anything like this before the Roman period. Of course, we know that this didn't happen throughout all Iberia, of course, but we know that this happened in more Mediterranean climate areas. So when we move to the more Atlantic northwestern Iberian sites, things are a little bit different. We have plenty of sites sampled. I think these maps probably are not updated. We have more samples, more sites in these ones by now. But we have a lot of sites, and they provide us some very relevant information. We have not all sites provide us with a lot of data, considering there are very large discrepancies in sampling strategies. However, those ones are improving through time. And what we find is, besides the naked wheat and barley that we find everywhere, like I mentioned before, we have this very relevant presence of millet, much more relevant here in Northwest than we find in any other region in the Iberian Peninsula. You know that millet increases. The relevance of millet tends to increase throughout the Roman period in other Iberian regions, but in the Northwest, it occurs much earlier. It is one of the most relevant crops during the Iron Age. We find it everywhere, and together with spelt, comestible spelta. So this is a hulled wheat, and it's probably the most relevant crop in Northwest Iberia during the late Iron Age, at least. It was introduced in the region in the Iron Age. So in the south, we have an introduction of those fruit trees. In the Northwest, we have spelt coming on. It is found in other places in Iberia, but it's clearly predominant in the northwest. And there's also the introduction of oat, or also in the Iron Age. This occurs probably around for the 5th or 6th century BC. So there's this new crop coming in the Iron Age. We have difficulty in knowing where the crop comes from, where spelt comes from. Spelt, of course, is originated in the Near East, but genetic studies are not very clear because it seems there is also a secondary origin somewhere in Central Europe. So we are not clear. We don't clearly know where our spelt is coming from. But we know that this is found everywhere too. It is, we have some particularly relevant sites where where spelt is found in the large amounts. These are storage areas that got burned. So we have a lot of material, and they seem to have been stored, partially processed in order to endure long-term storage. And so this is probably a very relevant for showing the resilience of these communities, particularly in this harsh environment of the Northwest. It was introduced in what seems to have been a very cold period in the Iron Age. Later on, there is another new crop that is rye, but the chronologies that we have suggested were introduced somewhere in the late Iron Age or the early Roman period, and it was first introduced in northwestern Iberia. The oldest chronologies we have are here in northern Portugal. And only later on it would spread through the rest of the Iberian Peninsula, mostly in the late Roman times and in medieval times. So it's something that came first to the Northwest Atlantic Iberian. It would spread, as I said, in the late antiquity, as we see in this incredible site of Monte Mozinho, and we have other sites suggesting that that somewhere after the first century crisis, rye ended up becoming a very relevant crop in some areas. Why are things different here in Northwest when compared to the Mediterranean regions is not completely clear. The first hypothesis, and I think it's probably the one that makes things more clear, is that there are major environmental differences. And that is true, of course. We know that spelt in the case of spelt is more clear than any other crop. Spelt is it's a very undemanding crop in terms of soils and so on, much less than naked wheat. So it is suitable for less fertile areas. However, it doesn't tolerate drought. So it is more yielding than naked wheat in case of mountain areas with high humidity, with very high rainfall. This has been demonstrated by agronomic studies in Italy and experimental studies in the UK. So in a region like the Northwest Iberian, where rainfall is quite incredible, I think it's one of the highest rainfalls in the whole Europe, at the level of the UK and something like that. So it's really something that people used to think, usually people think of the Iberian Peninsula as a very dry area, but not Northwest. So spelt would be particularly suitable for the mountainous and very humid environment of the Northwest. So this is probably the best way for us to interpret this predominance of wheat in often the spelt wheat in the northwest. Again, of course, there are major differences in terms of cultures. We know this. We know that there is this higher predominance of Phoenician influence more to the south, much less to the northwest, and here in the northwest we find what is usually called Castus culture. So whether this particularly cultural background, if this has an impact on crop selection, we don't know for sure, but it's probably something like that is occurring. And it continues throughout at least the beginning of the Roman period in the region, considering that, of course, during the Roman period, people continue to be the same as they were before the Roman period. So people continue to cultivate more or less the same things, at least in the beginning of the Roman period. So what we notice is that there are fig trees, or almost no fig trees during the Iron Age in Northwest Siberia. And why? Why are there no fig trees? It's not completely clear. If the expansion of fig trees occurs due to Phoenician influence in the south, well, we have clear evidences of Phoenician contacts and Punic contacts in Northwest Siberia. So these two worlds were completely in contact. However, there are no actual colonial sites in Northwest Siberia. So probably that is the reason why. We're not sure. Of course, the environmental issues are, again, a very relevant issue. So there are no fig trees, and probably there is a combination of environmental and cultural facts that could justify this absence in the Northwest during the Iron Age. In the Roman period, well, only in the Roman period do we find these fig trees in Northwest Siberia, most from two waterlogged sites like Glacial and Aqua Flavia, and one carbonized assemblages from Bracarel. So in Vigo, in the 3rd century AD, so already in the late Roman Empire, we have this massive assemblage that was studied by Teixeira, and it's been published, so you can find it easily, find it in the internet. It's a very interesting assemblage that includes the fruit trees like peach and cherry trees and chestnuts and so on. And this is the difference also that we almost don't find in the south, the chestnuts, but we find it here in the northwest. Of course, again, this is likely a more of an environmental issue. So chestnut is native to the Iberian Peninsula, mostly into the northern and northwestern areas of the Iberian Peninsula, and that is where its cultivation will start in the Roman period. So again, in the late Roman levels of Aqua Flavia, we find also some of these fruit. This is a healing spot that was destroyed probably by an earthquake in the 4th century AD, and we find several fruits there, like the chestnuts and peach and plums and cherry trees and Juguns regia also. Sorry, I forgot the name in English. And so, but these are all from late Roman levels. Only recently, we found some of these fruits in the 1st century AD Roman level in Bacca Augusta, and some cremation associated with cremation depositions. So this is a completely Roman context, where some of these fruits were incorporated in the pyres as a consequence of being eaten in funerary feasts or being incorporated as food offerings. Either way, this is a completely culturally Roman context. So this is suggesting that Roman cities may have had a very relevant role in the introduction of these new foods in the northwest Iberia. So, but this is occurring almost 9 centuries after it occurred in the south. And the scale of production is never in the northwest as it is in the southern area. So these mountain areas are not prone for great monocultural exploitation of olive trees or so on. So what we find is usually smaller farms with wine or olive presses. So they were producing wine and olive oil during the Roman period, mostly in more warmer areas. So we do find them, but not with the scale of production that we find more to the south. Wrapping all this up, you're probably already very tired of listening to me. So what we find, there is some great continuity since late prehistoric times in terms of cereals in the Mediterranean area, and a great diversity of pulses. I haven't talked about this, didn't have much time for it. And while in the Atlantic, we have, again, this continuity of late prehistoric package of cereals with a low diversity of pulses, contrary to what's happening in the Mediterranean areas. But we have an introduction of spelt that became a major crop in the Iron Age and in the transition for the Roman period. And we have the introduction of rye in the Roman period, or in the transition between the Iron Age and the Roman period, that only became a relevant crop in the late Roman Empire. Another major difference is occurring at the level of fruits production and fruits consumption. We have the introduction of Eastern Mediterranean species and expansion of native species like olive and vintage in the Iron Age in the Mediterranean areas in southern Iberia, and then strong expansion and specialization of production for long distance trade in the Roman period in the Mediterranean, while in the Atlantic, this introduction and expansion occurred only in the Roman period. And still, although we do find some of these species in Roman sites, we cannot be sure that they were in fact being produced in Northwest Iberia. Well, for chestnut, yes, for Juglans regia also, because we find the wood of these species. So we have the woods and we have the fruits. So probably we are sure they are cultivating it. But we can't say the same for peaches and plums. So we are not sure if they are really cultivating or if they are just consuming something coming from elsewhere. But still, there are clear differences between the two regions. And although there are some limitations regarding the preservation of these materials and the fact that we only have very few waterlogged sites that can provide more information on fruits, mostly fruits. Still, we can be quite confident that these changes, these differences still, they are probably true. They are not an artifact of investigation. So these differences, as I've been saying, are probably in relation with the broad environmental differences between the two regions, but also with the indigenous and cultural background of the two regions, and the fact that the Phoenician contacts were much more close and a wider effect in the southern areas. What we see, of course, is there's a major gap in information. We have large areas without sites or with very few sites with data. So, and this is a major problem. By now, we have a project going on. It's called the Be Roman. You can follow us on Facebook. And we are trying to fulfill some of these gaps by studying sites in southern Portugal and and in Spain also, in what was the Lusitania province. These are basically Roman sites, but some of them have also Iron Age levels. So if we're lucky, we can have some information that can connect these two areas, the northwest and the southern Spain. Probably 2 years from now, we may have some information that can help us understand where the border is, how these changes or these differences between these two areas, how can we find them in this middle area between the two. So if we're lucky, we will find enough data to help us understand this. And that was all. Thank you.
This was a last minute sort of change to our intended schedule for this afternoon. So I'll try and convince you all that genetics is interesting, and it can be used to answer important questions. As Carlos was saying, I specialize in human evolution, so I will not be talking about plants like João did before me, but I'll be talking about the encounters between modern human populations and our extinct cousins, in the particular case of the Denisovans, which are a mysterious group of extinct human relatives that inhabited the continental Eurasia, but mostly East Asia, between 200 and 50,000 years ago. So that's really, really long term. If you go back in the past, we're talking about deep prehistory in the Pleistocene. But because my talk is mostly going to focus on genetic analysis, I'll start by trying to give you a sort of crash course in population genetics and sort of the way we think about human evolution, or about the evolution of different genomes. It could be plants, it could be humans, it could be animals. Evolution is one process that affects all life on Earth. And so I'll just give you a crash course to try to get you into the mind of a population geneticist trying to understand how humans migrated across the world and how they encountered, and like how do we measure these encounters and admixture events between different groups that at the end led to the diversity of humans that we see today. So genetic information is stored in cells, right? We all have cells, billions of them, right? That's what we are made of. We're made of cells. Genetic information lives inside the cells, and we have different types of genetic information. Mostly when you hear about human DNA, we're thinking about the DNA that lives in the nucleus of the cell, right? So the cell has different sort of compartments. It's its own little universe. And so within the cell, you have the nucleus, and within the nucleus, we have the genetic code that sort of encodes for everything we are, from the color of our eyes to the color of our hair, how tall we are. So the physiology and metabolism of our bodies, right? And so the DNA is stored in little sort of packets or sort of bags of information that we call chromosomes, right? So you have a chromosome here, then you have several here depicted, and each chromosome is composed by this sort of coiled structure where you have the DNA sort of double helix, right? And so that makes your genetic code. The code itself is only made of 4 letters, right? It's A, T, C, and G. And those letters refer to the nucleotides, sort of the chemicals that make the genetic information of humans. And so you have, each letter corresponds to a different nucleotide, which are called adenine, thymine, cytosine, and guanine. It doesn't really matter. It's just that we have, imagine an alphabet with 4 letters, and the combination of those 4 letters give different books. So each human, each living individual, each species has its own book, right? Then we have what we call mitochondria. So these are like the batteries of the cell. So this is what produces energy that make metabolism possible. Mitochondria used to be living outside eukaryotic cells, and it was sort of through the engulfing of a bacteria into the human cells that we got these mitochondria that now we cannot live without, right? They are part of us now, but they used to be bacteria. They have their own genetic code, right? And it's a circular structure, much more simple, right? So when we reproduce and we transmit this information to our kids, we are transmitting mostly our own genetic code. So this is stored in chromosomes, but we also transmit the mitochondrial DNA to our descendants. So the human genome is composed of 22 pairs of these chromosomes, right? So imagine like these 22 bags, and you have 2 copies of each bag, right? One that you inherited from your father and one that you inherited from your mother, right? That seems straightforward. Now, the numbers 1 to 22 just refer to the size. How big are these bags? Imagine that you have little books and big books in your shelf, right? So imagine chromosome 1 is like reading a really big encyclopedia, and chromosome 22 is maybe reading the newspaper, right? But each has different information. Then males have an additional 2 bags, one called the X chromosome and the other one called the Y chromosome, right? Females have 2 copies of the X chromosome. So you can now already see that X chromosomes are inherited from our moms. So our moms always passes the X chromosome, and then our dads passes the Y chromosome if you are males, or the X chromosome if we are females, right? This is how the human genome is organized. If you look at the family, so if imagine this is a pedigree, and we have here a boy, and the reason we have a boy is because we can depict the Y chromosome, right? So we have a boy, and when we look back in the generations to see how the genetic information in this boy sort of got transmitted, we can see that these 2 bars, the bigger bars, refer to the 22 pairs, right? It's just 2 of each. So for simplicity, we don't put 22, we just put 2, right? Those are the 22 pairs. And then we have a Y chromosome, and we have the mitochondrial DNA, right? The mitochondrial DNA comes from the mother. So the boy inherited from his mom, who inherited from her mom, who inherited from her mom. So the mitochondrial DNA that each of us males carry has nothing to do with the mitochondrial DNA of our fathers, rather from our mothers. The Y chromosome, you can see, is inherited from the male lineage. So just think about when we're looking at the genetics of a human population, depending on which type of DNA we're analyzing, that's giving us different information about that population, right? And then you can see that on the big bars, which are called these autosomes, so each of these 22 pairs, you can see that there's different colors, right? And those different colors, they refer to portions of each different ancestor. So imagine this green portion here. It comes from the father here, who inherited from his mother, who inherited from her mother, right? And you can sort of follow through each segment of the DNA across the generations. So if you're looking at the 22 pairs of chromosomes, you're looking at the entire family tree. If you're looking at mitochondrial DNA, you're only looking at the female lineages. And if you're looking at the Y chromosome, you're looking at only the male lineages, right? Well, how does this work? So how do we actually make this work and think about this for thousands or hundreds of thousands of years, right? Evolution is like physics. So you have different forces. You have mutation, recombination, migration, genetic drift, and natural selection. That's as easy as this, right? If you know these 5 forces, you know exactly how your genome came to be. Mutation is just a process where the copying of the DNA is imperfect. So if you receive a copy from your father or your mother, it's not really exactly the same. Why? Because the machine fails, right? It's not perfect. And because it's not perfect, when you have spermatogenesis or the formation of eggs, what happens is that the copying mechanism makes errors. The machinery that we have that corrects those errors doesn't see that, and it's transmitted to the generations. And that's why humans are different between ourselves. We are different from chimpanzees. We are different from monkeys. We're different from tigers, and we're different from plants. We all share a common ancestor, but evolution through millions and millions and millions of years made all this diversity, right? So that's due to mutation. Then you have a sort of a more obscure force. It's called recombination. I already alluded to it. Recombination just shuffles genetic diversity from across your ancestors as you go back in time. And I'll go into that so you can see exactly what I mean. Migration, usually people think about migration as moving from place A to place B. Geneticists don't think about migration this way. We think about genes moving from genetic pool A to genetic pool B. So if you have a reproductive event between two individuals of different populations, we see that as a migration. They could be in the same place, but this is a migration event for us, right? Then you have genetic drift. Genetic drift is just the loss of genetic diversity due to purely random stochastic events, right? A meteorite falls in the earth, kills the dinosaurs, they're gone. It's not because it was good or bad, it just happened, right? So most genetic diversity is just lost through this. And then you have natural selection, which I think most people are familiar with, is when you have different genetic variants that are sort of better adapted to particular climatic conditions in particular places, in particular times. And so that variation sort of survives better. So individuals carrying particular mutations will survive and reproduce better than others, right? So that's natural selection. But today we'll focus on these three. And particularly, if you sort of get the next 2 slides, you'll get the rest of the talk. Otherwise, I don't know, just interrupt me and we can go through that. Right, so mutation, recombination, and migration. What is mutation? As I said, mutation is just this process where you have the chain of the DNA just being changed through time, right? So it's imperfect and it accumulates. So if you have mutations in the germline and you pass that sperm, sort of fertilizes an egg, that individual is not going to be exactly like their father or the mother, right? So it has mutations. The more time passes, so the more generations pass, the more mutations accumulate, right? Because there's just more chances for errors to occur. So if you compare the sequence of 2 humans, the number of mutations between those 2 sequences is smaller than if you compare the sequences in a human to a chimpanzee, right? So we're close to the chimpanzees, but we're not as close to a chimpanzee as we are to a human, right? Because why? Because there's more generations that passed since they last reproduced, right? So you can use the mutations between any 2 pairs of sequences to estimate how long ago they shared a common ancestor, right? So if you have a brother and a sister, they share the common ancestor 1 generation ago, right? That's their parents, right? They still have mutations between their DNA, but they are very few. If you look at cousins, they share a common ancestor 2 generations ago in the grandparents. And you can do this exercise for all the humans on the planet, and we're all the same family, right? So if you take then geographical partitioning of these populations, so if you compare populations living in Africa, say, to populations living in Europe, then you expect that the number of mutations that separate, say, Africans to Europeans is larger than the one separating Africans from Africans or Europeans from Europeans, right? That's how we sort of see how then human populations are sort of related to each other, right? And then you can extend this reasoning to Neanderthals, to Denisovans, right? If we would have admixed with chimpanzees, you could do this too, right? You can sort of see whether there was any admixture event between different populations or different species just by looking at the DNA, right? And sort of this is what's depicted here. You can take anybody in a population, compare their sequences and estimate how long ago they shared the most recent common ancestor. Now, don't make the mistake to think that there was only one person living at a time. Is that all of the other genetic lineages just went away. They became extinct. They didn't reproduce until the present day, all right? Cool, so what is recombination? Let's assume that we have chromosomes, right? In chromosome 1, we have 1 from dad here in blue and 1 from mom here in red. So let's say these are my chromosomes. When I'm gonna pass these chromosomes to my children, the 2 chromosomes in each pair, they're gonna pair up, right? That's what happens when gametes are formed. They pair up and they also exchange genetic information. It's like that every time sperm cells or egg cells are produced, the chromosomes themselves are like having sex, right? They're exchanging information. So what you're passing on to your children is not the chromosome that you got from your mother or the chromosome that you got from your father with 50% probability, but it's a mosaic of those 2, right? So what you're passing on is sort of this hybrid chromosome. You're not passing a pink chromosome, right? You're passing sort of a blue with patches of red or a red with patches of blue, right? So these are ancestry blocks and we can trace them through time, right? We can trace, let's say, this blue portion here, and we can see that this guy got this blue from his father, who got the blue from his mother, who got the blue from her father, who got the blue from his father, right? And you can sort of trace, you can see that if we call it the individuals in the parental generations in different colors, the current generation is sort of a mosaic of all the genetic ancestries that are in the past, all right? So this is important now. So now I'm gonna shift gears. I gave you a crash course on how we think about evolution. Now we're gonna look at ancient DNA. So what is ancient DNA? Well, it's just obtaining genetic material from extinct individuals or people that are dead. And this could be people that lived 50 years ago. This could be people that lived 50,000 years ago. This could be plants, right? We could take we could take João seeds and we can look at DNA on those seeds if we can. We can look at animals, right? It's just the ability that we take some sort of biological material, of really degraded and old. We take it to a specialized lab with clean facilities and we extract DNA. And then we do everything that we do with DNA from people today. It's the same thing, right? It's just the process of extraction takes a bit more caution. This was developed, Svante Pääbo is really the guy that has developed the ancient DNA techniques for the past 3 or 4 decades. There are other groups, but I put his figure here because I'm going to talk about today extinct human relatives. And Svante Pääbo's group was the first to sequence a Neanderthal, right? Neanderthals were a group of humans, if you want. That's how I think about them. They were a group of humans that split from sort of the modern human gene pool sometime around 600 to 700,000 years ago, right? And that's based on the fossil record of Europe and the calibrated dates that we got from the DNA, because Svante Pääbo's team was able to obtain DNA from Neanderthals. And what they found is that when they analyzed DNA from Neanderthals, they saw that, in modern humans, and they compared the genome of the Neanderthals to the genome of modern humans, they saw that modern humans carry these small patches of Neanderthal ancestry, right? So think about recombination again, and you have Neanderthals and humans admixing, and you have the hybrid generation, right? First generation, you have half of the chromosomes from Neanderthals and half of the chromosomes from humans. As time goes by and these people keep sort of breeding with one another, then you start having these patches of blue and gray sort of mixing, and you have this hybrid sort of mosaics of ancestry. So each of us now in the room, if we're all Europeans, actually everybody, if we're not African at least, we have Neanderthal patches. So in blue, we have in our chromosomes Neanderthal ancestry that is a result of those encounters between modern humans and Neanderthals when our species first left Africa, some 50,000 years ago, okay? But they also did something else. This team in Leipzig did something else. In 2011, they went to this cave in the middle of Siberia, and they found a really tiny bone. So that is depicted here in this photo. So that's a small fragment of a bone from the pinky finger, right? And some archaeologists, this was definitely not a geneticist. Somebody looked at this and said, wait, this is a bone. This is a human bone, right? Looks like a seed, right? I was looking at João's seed and I was like, yeah, well, that could be the Denisovan bone. So they looked at this bone and they tried to extract DNA. And what they found was something amazing. They found that this bone belonged to a human group that was neither modern human, so it's not the same sort of species that we are today. It was also not a Neanderthal, but it was a sister species to the Neanderthals. So something that was as related to the humans as Neanderthals were. Moreover, so this is just a picture showing you where that bone was found. So that bone was found here in the Denisovan cave in the middle of Siberia, right? We have Neanderthal fossils all over Europe. And we knew that Homo erectus has lived in East Asia, right? So here in gray, you have the sort of the range of Homo erectus. But what we found, or what they found, is that this Denisovan individual that was found here was very close to Neanderthals to be Homo erectus. And the problem is that, according to the fossil record, Homo erectus had split from humans 2 million years ago, right? So if the Denisovans were 700,000 years ago, that's when they are estimated to have split from humans. So they're too recent, to be Homo erectus. So the question remains, who are they? And they were firstly described in 2011, and still today, we don't know who they are. Their identity is one of the big mysteries in human evolution. So in this map, I just want to show you as well that sort of like, imagine this was the world somehow, maybe 200,000 years ago, right? 200,000 years ago, we had humans in Africa. We have Neanderthals in Europe who had left Africa, right? Maybe 700,000 years ago. We had Homo erectus in Asia and in island Southeast Asia. And I'm going to talk about island Southeast Asia very soon, right? We had Homo erectus here, left Africa maybe 2 million years ago. And we have another group, which we don't know. We can't match. Because there's only that pinky finger, we have no morphological characterization of this species, right? So we don't really know who they are. What they also found out is that these Denisovans, who were firstly found in Siberia, they had admixed with modern humans. And the modern human populations, present day human populations with the highest amounts of Denisovan ancestry were found in Australia, New Guinea, and the islands of Indonesia and the Philippines. Right? This is really far away from Siberia. And interestingly, populations in East Asia, while carrying some Denisovan ancestry, that proportion in the genome was much lower. So that sort of points to separate events of admixture between Denisovans and modern humans, somewhere when our ancestors arrived in island Southeast Asia. Now, you think this is a bit mysterious, it gets worse. I mean, we know very little about these guys. So now I'm going to talk about island Southeast Asia and how does this Denisovan individuals get into the picture. So island Southeast Asia is a really, really, really interesting place for several reasons. So biogeographically, it is located, and I'm going to talk about the islands, right? So we have Southeast Asia, continental Southeast Asia, and you have Southeast Asia, island Southeast Asia, okay? So I'm showing you a map of a sea level reconstruction of the last glacial maximum. I know it says here 50,000 years ago, but this was 20,000 years ago. Now you have to trust me that 50,000 years ago it was very similar, right? What you have is that the current islands of Borneo, Java, Sumatra, they were connected by land to continental Southeast Asia, right? To where today you find Cambodia, Vietnam, Laos, right? Malaysia. These were all connected by land. And so when modern human populations arrived from Africa, they would have found this region completely connected, right? This was part of the continental shelf. Why? Because due to the ice age, the sea levels were lower, all right? So you had lowered sea levels. But then you had the islands immediately to the east of this region, which were never connected to the continental shelf. Why? Because of this thing called Wallace's line, which is one of the world's largest biogeographic barriers for the dispersal of terrestrial fauna. So animals, humans have tremendous difficulty crossing this unless they really intend to. Why? Because the sea levels here, so you have a maritime current that is very strong, that goes all the way, circumvents the Philippines, comes all the way down through Sulawesi and Borneo, and then between the islands of Lombok and I think Bali. And it's the due to the seismic activity and like the differences in the continental shelves, the continental shelf in the west is much higher than the one in the east. So you have this really deep trench where the water just forms strong currents. So if you try to swim or try to go by boat and row from one island to the other, you might end up in Australia or in Antarctica, right? So it's extremely hard to cross. And so we know that this has been there for at least 50 million years. And this has been responsible for the separation, for instance, of marsupial mammals that we find today in Australia and in New Guinea from what we are, placental mammals, right? So this is as deep as that. But we know that for at least 700,000 years, humans have lived in these islands. So we have, first of all, west of Wallace's line, we have stone tool evidence and fossil evidence for Homo erectus for at least 1.5 million years ago. We have stone tools in Sulawesi 200,000 years ago, in Flores and in the Philippines, maybe 700,000 years ago. But we also have two different fossil groups. Homo Luzonensis was described in the Philippines only in 2019. No one knows how close this species is to the modern humans. We have Homo Floresiensis, who has been described in 2005 or 2004, also in this area, in the islands of Flores. These two groups represent human populations with very, very tiny statures. They were about 1 meter tall, right? But because of the preservation issues associated with the tropics, right, it's very hard to obtain DNA from these fossils. We don't know how these species are genetically, right? So we know the age of the fossils. Most fossils are about 50,000 years ago, but the earlier evidence is about 700,000, right? we can look at them morphologically and sort of compare to other groups, but we don't know genetically how close they are to us, right? And now, if you look at, again, the Denisovan ancestry in this region, right, this makes it extremely interesting. So you have stone tool records, you have Homo erectus, you have Homo Luzonensis, you have Homo Floresiensis, but present-day populations carry DNA from another group, which we don't really know what they are or what they look like, and we found them in Siberia. So how can we sort of try and put this all together? It's very hard, right? So our question, and that's what I'm going to talk about today very briefly, was, can we sort of find evidence of whether these two groups, either Homo Floresiensis or Homo Luzonensis, admixed with humans, right? So we call them hominins, and that's why the title of this talk is Hominin Admixture, right? We want to find out whether these two groups, which we don't know anything about genetically, admixed with modern human populations, and their DNA presently survives in Indonesian and Australian populations, right? So this is what we tried to figure out. So imagine that, again, we have a human chromosome, like I showed in the beginning. This is shown this by blue bar, right? And then on top of this human chromosome, this individual, let's say this is an individual living in Indonesia today. This individual carries Neanderthal and Denisovan ancestry, right? So the small blocks that I've talked about, right? They are shown in red. And let's assume that maybe they had this super archaic, and super archaic just means really, really divergent group, right? It's just the Neanderthal and Denisovans are the archaics, the Luzonensis and the Floresiensis are super archaic. It's just a measure of how distant they are to us, right? Let's assume that they also have this super archaic admixture. But there's no way we can tell, right? Because we don't know what the genetics of these two species was, because we cannot extract DNA from them. So what we did is that we compared, so we first identified these segments that are Neanderthal or Denisovan in present-day individuals, right? In contemporary populations. And then we use a different method, right? That takes some of the stuff that I told you about, that we know from evolutionary theory and population genetics, and we use the new statistical method that was developed by a guy called Loritzkov in 2018. And the method simply does this. It goes along the genome and says, wait, I found some region that has a lot of mutations compared to a reference human population. So imagine that I take my DNA, right? And I don't know anything about Neanderthals, right? And I just compared to the same region with somebody else. And I found that these two individuals are mostly the same across the genome, but there are some regions where they are really, really different, right? And that may be because those regions in one of the individuals were inherited via the crossings with archaic species, right? Why? Because of mutations accumulating through time. Since two species split, when they come together and that DNA passes from species A to species B, in species B, those segments are going to be highly, highly diverged, right? They are going to carry a lot of mutations. So without knowing the genome, I can sort of suspect that those regions came from admixture events with other species, right? So this is what we try to find out. And then, of course, there are errors, right? I mean, because I don't know what the sequence of this of these species are, I'm just like guessing. Oh, this looks really unusual. Think about it in this way, right? And we've done this, we've applied this to 400 genomes, human genomes from island Southeast Asia. And this was based, or human genomes across the world, sorry, including more than 160 from Indonesian, island Southeast Asia. And these individuals were obtained from after a paper published by two colleagues of mine, Guy Jacobs and Mary Cox, and also the collaboration with Erawati Sudoyo at the Eichmann Institute for Molecular Biology in Jakarta in Indonesia. And so we use these three different methods. We identified Neanderthal regions, the Denisovan regions, and sort of putatively something else, right? The cool thing is this new method also gives us the Neanderthal and Denisovan regions, right? So we sort of can confirm that whether it's doing relatively well or not. What I then did is that we sort of focused and tried to estimate the amount of these residual sequences. So how much of the DNA of present-day individuals is suspiciously old or is suspiciously divergent that could be the result of admixture events with other species, right? And so the first thing we did is that we looked at the proportion, how much of the DNA from these individuals comes from Neanderthals and or Denisovans, and potentially something else, right? And this is what I'm showing you here in this plot. So the y-axis is just the amount of DNA, so the amount of sequence, and the x-axis is each of the populations we analyzed across the world. And it goes from west to the east. You have West Eurasia, so these are Europeans, Siberian populations, South Asia, these are mostly Indian populations, East Asia, so like China, America, so we're talking about Native Americans here, Southeast Asia, so this is continental Southeast Asia. Then we go West Island, Southeast Asia, so these are regions west of Wallace's line and populations living there, and east of Wallace's line in East Southeast Asia. Papuans, there's populations from New Guinea, and Australians, right? And so if you look at the amount of Neanderthal and Denisovan ancestry in each of these populations, each dot is a chromosome. So each individual has 2 dots. If you look at the distribution, you can see that the amount of Neanderthal and Denisovan is sort of similar between populations until you go east of Wallace's line, and that jumps, right? And this is what I told you before, that when you go and you look at populations east of Wallace's line, due to the higher amount of Denisovan ancestry, you have the total archaic ancestry increase. But if you compare to the unknown, these residual sequences, then we find a much lower amount of putatively admixed regions from superarchaic, so say from Homo Luzonensis or Homo Floresiensis. So this is already telling you that maybe there's nothing really to find out here. Okay. What we then did, I hope you're still with me, is that we looked into these residual sequences, right? So we say, okay, we understand these are Neanderthal and Denisovan sequences here, we identify them. Now we're going to look at the other ones, right? We don't find a lot of them, but how qualitatively, how are they? And we know again that mutations accumulate as a function of time. The more time passes, since the divergence between 2 individuals or 2 species, the more mutations are separating them. If we assume that Homo Floresiensis is really highly divergent, or Homo Luzonensis, and they are related to, say, Homo erectus, right? Then they split from humans 2 million years ago, or close to that, right? So we know that in this bar here, there's 2 million years of evolutionary time. And then we know that Neanderthals and Denisovans split from humans around 700,000. So that's another 1.3 million here. So if I sum up 1.3 with 2, I get 3.3 million years of evolution that could have resulted in mutations accumulating in the DNA of Homo Floresiensis that I don't see in humans, Neanderthals, or Denisovans, right? So this could result in an excess of positions, of nucleotide positions in the genome when I look at the code, where if I have an admixture event and Homo Floresiensis did indeed admix, say, with the ancestors of Papua New Guinea populations, right? When humans arrived in island Southeast Asia, maybe these populations carry an excess of mutations that are different from Neanderthal, Denisovan, and other human populations, right? And let's code these positions as 1000 or 0111. That means that it doesn't matter. It's the sort of the ancestral or derived allele. It doesn't really matter. So you can have sort of these motifs, right? Where if I got DNA from a really divergent species, then I will have mutations that nobody else has, right? Because those are mutations that occurred in the evolution of that species that has admixed to form me, right? Does that make sense? I hope so. So we then look at the proportion of these mutations in the different populations, again, from west to the east, right? And what we found is that there are slight differences between these populations, right? So we have some populations with a higher proportion of mutations of this type. But we also found that these type of mutations correlate with how much Denisovan or Neanderthal ancestry these individuals carry, right? And that made us suspect that what we are finding in these residual sequences might not be really the result of admixture with other species like Luzonensis or Floresiensis, but rather Neanderthal or Denisovan sequences that our methods are not able to detect, right? So our methods are not perfect. So we're not going to detect everything that came from Neanderthals or everything that came from Denisovans. So if there are some regions that go undetected, then I expect that the suspicious regions are correlated with the amount of Denisovan Neanderthal that I do find, right? And that's basically what this is telling you. And then we did computer simulations where we used 4 different models, right? If we assume that there was admixture between Floresiensis, Luzonensis, and humans, and this was like, there was no admixture, so 0%, or very little admixture, so 0.1%, or 1%, or 2%, right? And we simulated this in the computer, right? So we made computer simulations saying, if I have this mutation rate, and I have this amount of generations, and I have this admixture occurring 50,000 years ago, what is that I expect in terms of the proportions of these mutations? And interestingly, what we find is that between the models between 0 and 0.1% of admixture, so basically nothing happening, are the ones that agree better with our empirical observations, right? So if you look at the proportions, say, of 1,0,0,0, we find between 23 and maybe 26%. And that's exactly what you're seeing here in the y-axis in the empirical observations. So overall, we don't really find any evidence that these 2 groups, these 2 fossil groups, which we don't have any genetic evidence for, contributed to the genetic ancestry of modern human populations. But isn't that weird? I mean, we have the Denisovan ancestry in this region. We have 2 fossil groups. We test whether if these 2 fossil groups are really, really divergent, whether they could have admixed. We find really no evidence. But so we don't have a Denisovan sort of fossil to compare. The fossil records for Denisovan is very poor. I mean, there's the pinky finger that I showed you, and now there's the skull that was found in China that people believe represent Denisovans. There's the mandibule that was recently found also in Tibet. But if you ask a morphologist or a paleontologist to ask, and if you ask them how is a Denisovan supposed to look like, no one really knows. I guess the assumption is that because they're genetically close to Neanderthals, they should look more or less like Neanderthals. But I mean, who knows, right? So the question is, these fossils are in the right place at the right time to be these mysterious group of Denisovans that were first described from a pinky finger bone in Siberia, right? So this is where human populations today have the highest ancestry. Of the highest Denisovan ancestry. So there's two possibilities here that are quite provocative from our study. One is that they are indeed the Denisovans, and I was sort of called heretic for proposing this, right? The other is that if they are not the Denisovans, then, I mean, if I were an archaeologist, I would really dig in this area because you have to find them. They have to be there. And if you find them, I mean, you're going to be able to describe, morphologically a new species of humans. So the conclusion is there's a big finding coming our way. Doesn't matter how you look at our results, right? I personally suspect that they are the Denisovans. We're just too conservative because morphologically they are weird. They are small. They have archaic features in their bones. They look more like erectus, but I mean, who knows? I mean, evolution in islands can do really weird things. We know elephants became dwarfs. We know rhinoceros decreased their sizes. We know lizards can increase their size tremendously. I mean, this Komodo dragon, right? So what we know about the evolution in islands is to me, perfectly compatible with the fact that these guys could be the Denisovans. So who knows? There's other possibility, which is that everywhere we find fossils in these islands, and there's 2 places essentially. Everywhere we find fossils, we found that after modern human arrival, these islands carry still to this day megafauna. So that by megafauna, I mean animals that are larger than 50 kilos or that are heavier than 50 kilos, right? So most animals were wiped out. So in Australia, you don't really find lots of megafauna, right? Why? Because the idea is that when humans arrived, the hunting pressures just wiped out most of the species. But you do find megafauna in the Philippines. You do find megafauna in Flores. And there's another island where you do find megafauna. And that's Sulawesi. And Sulawesi is a really big island. Let me try it with my mouse. Yeah, it's here. It's a really big island. These look tiny in the map, but these are really huge. If you put Portugal there, it's going to look very tiny. So Sulawesi is potentially another, if the Nisovans are yet to be found in island Southeast Asia, Sulawesi is a prime candidate. If you believe that megafauna survives due to the consistent hunting pressures of hundreds of thousands of years of these animals being sort of pursued and killed by human-like groups, right? So this is a possibility that was, this is not my theory. This was put forth by Chris Helgen, a collaborator of mine and good friend at the Australian Museum. So we did put this in the paper, right? This is a possibility. So if you believe the Nisovans are yet to be found and you don't believe me, then Sulawesi might be your bet. All right, and that's pretty much what I had to show. I hope it was not too much of a genetics, geek knowledge, but I tried my best to explain as best as I could. So I want to thank all my collaborators that contributed to this paper and also people that provided comments, and and thank you for listening. And that's all.